So now we are going to move to adaptive immunity. So this is the adaptive immune process, and then we'll point out places where there's like interplay between the innate immunity and the adaptive immunity. So first, we need to talk about APCs. So APC stands for antigen presenting cell. I like to think about it as a professional cell that can present an antigen. So that's why this cell here has a tie, because it's a professional. So this professional cell has a few different features that we're going to talk about. So first, let's talk about MHC and like ways that we can signal like what is self and what is non-self. So all cells, first of all, have MHC1 and like HLA. So you can see information about this on slide 10 and slide 12 here. So like MHC1 is on all cells, all nucleated cells. So there are some exceptions like red blood cells, for example, because they don't have a nucleus, but all other cells will always have MHC1. And we'll see this come into play a little bit later. So all cells have that, but then also these cells have, like these specific cells, so APCs are special because they're professionals and they've gone to school and got their license to be an APC. And so they have MHC2. So like that means that an APC, they have MHC1 and they have HLA and MHC2. So they have all three of those. And so this here is like our MHC2 or our license to be an APC. And then so APCs are cells that are like phagocytes. So let's talk, before we see exactly what happens here, let's talk about the kinds of cells that can be APCs. So we have, let's see, what is our color coding here?
Macrophage.
Okay. So the macrophages can be, and remember monocytes turned into macrophages before. So macrophages can are phagocytes that can be APCs. This one is going to be
B cell.
So B cells, which are parts of the adaptive immune process themselves. And then lastly, we have my favorite for some reason of these ones. I really like these guys. That are the dendritic cells. And so we did not talk about these before, so we'll talk about them a little bit more. I'm gonna spell this wrong.
TIC, dendritic.
Yeah, okay.
Perfect.
Okay, and so dendritic cells are these cool little guys that they also live in the tissue. So before we talked about the only cells that like live in the tissue and normally just hang out there were like the mast cells, but mast cells aren't phagocytes. Dendritic cells are out living, they live in like the tissue. So like where we were before with like our innate immunity, when they're just like out in the tissue surrounding the blood vessel also are like the dendritic cells. And these guys are also professionals, but like they are phagocytes. They can go and eat like pathogens or cellular debris, and then they take them and then they will come into play here with everything else. So the only difference between dendritic cells, macrophages, and B cells is that these inherently always live in the tissue, but they are mobile and they can get out and like move into circulation to interface with the adaptive immune system. So these are the cells that can be APCs. And then lastly, what will happen is that, so like for example, with our dendritic cell, we'll say that like he's right here. So like this is our dendritic cell. We know that he has MHC1 and HLA. He also has MHC2. And then, so when he goes and there is a pathogen right here, so this angry little pathogen, he is going to phagocytose this and eat him up, chew him up, and then process him. And then it's gonna take some of this guy and put it here. So okay, so that's just like a little piece of this guy. And so then we take the antigen on the MHC2, and like that, the APC will take that to go interface with the rest of the story.
Okay, so Ryan walked us through how antigen-presenting cells work, what types we have, and this is the beginning of our adaptive immunity. There's kind of two parts where the story kind of bifurcates into two different parts. We have the cell-mediated story, which is involving mostly our T cells, and then we're gonna shoot down to the end and talk about humoral immunity, which is more of a B cell themed story. So starting here, I'm gonna talk about the cell-mediated story. So for antigen-presenting cell, it's likely a dendritic cell, going around, taking in pieces of its environment, taking in pathogens, eating them up, and presenting the chunked up piece of the pathogen, which we're calling an antigen. So just a reminder, here's our MHC class 2. So the antigen presenting cell takes in the pathogen, eats it up, presents a chunk of it, what we're calling an antigen, on the MHC class 2. Now, this APC is gonna come and migrate to a place that is rich in T cells. So in this example, we're gonna say the thymus. The thymus is a place that has a ton of T cells. So this APC is now in here. It's in this place that has a bunch of T cells, and this T cell has the ability to recognize that kind of pathogen that our APC had just eaten. So the APC is gonna hook up with the T cell on the T cell receptor with the MHC class 2. So you can see this is how this connection is being made. Antigen presenting cell comes to a T cell rich area. There's a billion T cells here. This one happens to have the right shape and have the right recognition to hook up with this type of pathogen that I just ingested, the APC just ingested and presented. Now they've hooked up. Once this process happens, we move here. We're gonna see the T cells proliferate and differentiate. Proliferate means multiply, make a bajillion of. Differentiate means to turn in to different types of T cells. So T cells, after they've proliferated, they've multiplied, made a million, then we differentiate into CD4, CD8, regulatory T cells, and memory T cells. We're gonna call these T helper cells. We're gonna call CD8 cytotoxic T cells. So after we've differentiated, now we have these even more specialized T cells that are specialized to our angry pink pathogen. Now, let's say that we're gonna kind of pause this CD4 story for a moment. We're gonna go to this CD8 cell, the cytotoxic T cell. Kind of a backstory we need to understand that we kind of talked about a little bit is that self cells have MHC class 1, and they're presenting little signs to the community that they are self. So let's say in my body, it presents a little like redhead. And it's like, this cell is part of Abbey. This is part of Abbey. It's saying, I'm good, I'm in the clear. Now, if this cell was infected by a pathogen, then the signal on the MHC class 1 would look like me, but maybe have like pink hair. So it's trying to pretend that it's okay, but a CD8 cytotoxic T cell that comes by is gonna recognize that this is not self. It's gonna say, hey, usually this is the signal on your MHC1 is a nice, cute little redhead, but this is not right. And this cytotoxic T cell is now going to destroy this self cell because it's infected with a pathogen, some sort of virus. So it does that by releasing perforin, poking holes, and then this cell will explode. We're okay with a self cell exploding because this self cell has been infected by a virus or by a pathogen. So we need this cell to be terminated so that it doesn't keep continuing to multiply and replicate and keep spreading that. So T cells, they differentiate. That we have some memory cells that help us remember certain pathogens. We have regulatory T cells that help keep our immune system from overperforming. We have the CD8 T cells, which we're calling cytotoxic T cells, that are gonna recognize bad signals on the MHC1 and destroy it. And then we've got these CD4 T helper cells, and we're gonna pause here for a moment.
Okay, so one thing that's important to differentiate between the two of these two parts of adaptive immunity. So when we talk about cell-mediated, like the cell-mediated story or the cell-mediated response, we're talking about things that involve the T cells. And so like the T cells are gonna be responding to things like intracellular, like viruses, because viruses need to go into cells and then replicate their DNA. So that's happening inside a cell. So so they are going and killing self cells, or like a self cell that has cancer, or like a self cell that has an intracellular parasite. Like any of those would be like things where like the T cells are going to be going to like go deal with that. So then let's contrast that with humoral immunity. This is obviously like a really dated term, but the humors refers to different things. But like one of the things it's talking about is like with blood. And so if like for things like if we have like bacteria, like if a patient is septic and there's bacteria circulating in their bloodstream, humoral immunity is going to help to deal with that. Also, if we have like let's say like bacteria that is like in a tissue, because like you've got like a cut and there's like bacteria in the tissue, the B cells will be dealing with that. They'll go into the tissue. They're not going inside of the cell, mind you, but they're going inside of the tissue. So humoral immunity is dealt with like with the B cells, and then cell mediated is like our T cells. So just wanted to clarify on that. Again, note our slide numbers so you can see like where in the slide deck we're talking about. So for our B cells, so like the same thing as before, we have our little our APC is going to come here. Our APC is like, you know, booping along, coming from over here. Yeah, so our APC is there. He's going to be coming over, and then so we'll just pretend in this one it's going to be like a macrophage here. So we've got our like macrophage, and then he's got his, we're really trying to drill this into your heads, so take note of that. This is MHC2, and then we have some antigen, which at the moment we're going to represent with a different color.
Black.
Okay, yeah. And so then this is antigen from a bacteria that like this macrophage. The macrophage ate a bacteria. It like processed the leftovers from that, and then put like the antigen from the bacteria onto its MHC2, because remember, it's a professional. So okay, so then this is our little APC guy. So then really this part would be pointing over here toward him, but that's neither here nor there. So but we're basically like the APC is going to come to a B cell rich area, like the lymph nodes, for example. And so it's going to go there, and then it's just going to basically be going and like looking and being like, do you have my perfect match? Do you have my perfect match? Do you have my perfect match? Going from like cell to cell to cell, which sounds exhausting, and that's because it is this first time that it's doing it. And so then it's going to try and find its perfect match. When we find a B cell, so a naive B cell or unactivated B cell that has a antibody, because they have antibodies bound to their plasma membranes, when it sees one that matches perfectly the antigen from that bacteria, then we have a match. And that seems like a great time for like celebration. But before like the magic can happen between these two, where this guy can start to proliferate, we need someone else to sign off on this transaction to make sure we're not going to start making antibodies to our own self cells. So this is where our friend and the ever helpful helper T cell comes into play. So he is going to come here. So this is going to be this is like a T cell, and then he's going to check. So you got like, he's checking it out to see like what, like make sure that like this, the antibody for this B cell matches perfectly the antigen from the pathogen. And then when it sees it, it agrees that like we have a perfect match, then it's going to sign off. It's going to sign off on that transaction by releasing, it's going to release some inflammatory cytokines, which you remember from innate immunity, of course, now. So these inflammatory cytokines will tell the B cell now that it can proliferate and differentiate. So the B cell is going to, we're going to come over here. So I mean, in essence, so we have this one, and then it's going to start making a whole bunch of plasma B cells that like have that perfect match. So it'll be, so now we're making a bunch of plasma B cells. And what do plasma B cells do?
Antibody machine.
Yeah, they are like antibody factories. Like literally, they can make like one plasma B cell can make, I think it's like thousands, like thousands of antibodies like per second. So it's a lot. So they're actually pretty impressive. So these are going to be our plasma B cells. So it's going to make those, and then we're going to talk about like the role of antibodies and what they do. But also our like B cells are going to differentiate. So like our B cell from like before. So then he can like, it's going to be making these like plasma B cells as one part of this. And then also we're going to make memory B cells over here also. Because in the future, we don't want this process to take so long. Because like normally, this process that we're describing where it's happening for the first time, it can take like, for us to get here where we've like differentiated and we make all these like little antibody factories, we're having like 10 to 12 days. So like you've been sick for a long time at this point. But if we have these memory cells, so in the future, now these guys will be out in like circulation. And so then if we get exposed to that again, they can recognize this, and then we like the time to being able to like start making a bunch of antibodies is very short. So like the memory cells will have that function. These are our antibody factories. And then let's talk about like the antibodies themselves. So you do still need to go and look at your slides, because we have like the like IgG, IgM, like the different like IgE. There's different kinds, and you do need to know those, and you are responsible for that, and you would be wise to at least like be familiar with those. But I just want to talk about like because obviously these B cells, they're not killing anything. So these B cells aren't killing anything directly on their own, but like their weapon of choice is actually pretty powerful, which is like the antibodies. So the antibodies are represented by these like little Y-shaped guys. And so these antibodies can do several things. So like they will, when antibodies are released, they will go to a pathogen, and then they'll start attaching to them. And when they are attaching to them, this is going to flag them for destruction by our phagocytes. So this is going to be
neutrophil.
Yeah, well, so like, yeah, I mean, not that part, but the word I'm looking for is opsonization.
Oh, yeah.
That's probably spelled wrong.
That's fine.
Yeah, so opsonization, so that's basically flagging like the pathogen for destruction, and so that'll be with like our neutrophils, our macrophages, and our, I think, probably like natural killer cells like can also like play a role in that, or like the dendritic cells.
Yeah.
So then here, this is going to be our antibody antigen complexes that we talked about with the complement, and so then that like tells the membrane attack to come and like kill directly by drilling holes in the pathogen, and so water rushes in and it will lyse the cell. And then lastly is just neutralization. There's a couple other things it talks about. I wouldn't worry about knowing them. But like they, but for the other, it's just neutralization. And so that's just going to be basically where we like just cover the pathogen completely in antibodies. And so then like the pathogen is just basically like, it can't do anything because it's surrounded by a circle of by antibodies. So like it can't go and like wreak havoc by like causing problems with your own cells, because we basically, anything that it could do anything with, we've just neutralized it. So like that last one is just neutralization.
Amazing.